UCanBeHealthier TV channel. I'm your host, Dr. Mamatha Chitriki, and today I'll talk about a scientifically proven way to freeze aging and add quality years to your life, like flipping a youth switch. This special video is intended for anyone over the age of 40. Today, I'm going to explain how to turn off the aging process by activating a newly discovered youth gene found inside our DNA. The key is living longer and enjoying those years in excellent health. Getting old and sick is not predetermined. You have an ironclad way to slow or even stop the aging process at any age. The ability to defy age is built into all of us. We just need to activate it properly. We all have a youth gene that keeps us young, but only 1% know how to activate it naturally. Unactivated, you feel old, get old, feel sick and suffer. Activated, you feel youthful, strong and healthy. Activation is what you will learn from this video, and once you know the secret, anyone can do it. Activated, you will feel strong and youthful well into your 60s, 70s and even 80s. Imagine how great it would be to look and feel decades younger without any drastic lifestyle changes. Maintain a 35-year-old body at 70, 75 and even 80 years old and beyond. Feel youthful in your retirement. Imagine your middle age being 70 or 75. Imagine staying active, nimble and never in discomfort. Imagine no longer having to fear the doctor, as you know that your health is solid. Imagine battling hair loss and tooth decline, building muscle and losing fat into your golden years, or supporting your vision and hearing like you were decades younger. Wouldn't this be fantastic? The secret is the SIRT1 enzyme and the ability to activate it. Anti-aging proteins are called SIRT, and they are responsible for the production of sirtuins. Sirtuins are proteins expressed by SIRT genes that perform an enzymatic activity stimulating chemical reactions essential for the body. Their function has been ascertained by several studies, although there still seems to be a lot to learn about them. SIRTs, in short, are proteins with enzymatic properties. They regulate the metabolic processes related to insulin resistance. They have control over immunity. They have a fundamental role in epigenetics. They're involved in defenses against cancer, and they're essential proteins that allow the cell to live longer. Scientists have begun understanding how to stimulate them pharmacologically and with natural molecules. When we are too stressed, it's often necessary to stop, take a breath, and regain energy. This rule applies not only to us, but also to our cells. If they are subjected to stress, our cells rest and renovate themselves. Natural evolution has selected precise mechanisms that allow the cells to enter a state of standby, a recovery system that prolongs the cell's life and that of our organism. But how does this work? Scientists have identified a family composed of 7 genes that play a fundamental role in this recovery system and are considered the most critical longevity genes. They are called SIRT genes and are necessary for the regulation of the metabolic processes of the cell, in particular responsible for its survival in risky conditions such as low temperature, physical stress, and fasting. These 7 longevity genes are responsible for the production of sirtuins. Sirtuins are proteins expressed by SIRT genes that perform an enzymatic activity stimulating chemical reactions essential for the body. Their function has been ascertained by several studies, although there still seems to be a lot to learn about them. Sirtuins depend on NAD+ or nicotinamide adenine dinucleotide, the coenzyme needed by every cell in our bodies. However, NAD+ levels naturally decline with age, and sirtuins functioning also declines. Maintaining NAD+ levels can reduce the decline in sirtuin function and minimize cellular aging and chronic diseases. Although you can't supplement directly with NAD+, you can take one of its precursors such as NMN, nicotinamide mononucleotide, to boost it. In addition, sirtuins can be activated by caloric restriction, exercise, transresveratrol, and curcumin. Each of the 7 sirtuins behave slightly differently, but have the same goal in regulating cellular health and controlling aging. Increased sirtuin activity has been linked to increased lifespan in yeast and animals and reduced risk of progression of cardiovascular, metabolic, and neurodegenerative diseases in humans. Some of the main functions of sirtuins include repairing damaged DNA, which is thought to be a leading cause of cellular senescence, upregulating mitochondrial activity, increasing antioxidant pathways, which reduces oxidative damage, responding and adapting to cellular stresses, and regulating telomere activity. Telomeres are the end caps of DNA that protect our chromosomes and shorten with age. Sirtuins also are involved in generating cellular energy through ATP production. So sirtuins are full of important actions. As NAD levels decline with age, sirtuin activity is reduced, which impacts cellular communication and contributes to the pathogenesis of aging and age-related diseases. Although sirtuins were only discovered approximately 20 years ago, the research is ever growing as scientists aim to determine how sirtuins impact human longevity. Initial research into sirtuins was done on yeast and animals. A 1999 study published in Genes and Development found that overexpression of SIRT2 extended the lifespan of yeast by 30%, fueling the idea that sirtuins promote longevity and reduce age-related diseases. Additionally, a 2013 study in Cell Metabolism found that increasing SIRT1 in the brain led to delayed aging and extended lifespan in mice, adding credence to the idea that sirtuins are involved in brain health. However, research on the sirtuins' impact on longevity has mainly been confined to yeast and animal studies. The primary correlation involves SIRT3. Single nucleotide polymorphisms, SNPs, in SIRT3 have been linked to increased human lifespan in centenarians. Although these results are promising, it should be noted that further research is needed on humans. Although SIRT2 was the first sirtuin to be discovered, SIRT1 is the most commonly studied of the whole lot. The reduction in SIRT1 levels, widely seen with age, is linked to dysfunction in the arteries, liver, blood vessels, endothelial cells, and brain. However, all 7 sirtuins play an essential role in health and longevity. The top researched areas in which sirtuins have been studied for their actions in human health are related to cardiovascular, metabolic, and neurodegenerative diseases. Cardiovascular disease. The leading players associated with mitigating the age-related decline in cardiovascular health are SIRT1, 3, 6, and 7. These sirtuins have been found to protect cardiac muscle cells from oxidative stress, suppress cardiac hypertrophy, and reduce dyslipidemia and the stress response in the heart. Due to the high energy requirements of the heart, the sirtuin's ability to increase mitochondrial function and ATP production is a primary mechanism in which they protect the heart. Of the sirtuins, SIRT1 and SIRT3 likely prevent cardiovascular disease the most, as they reduce oxidative stress and mitochondrial dysfunction, whilst protecting the endothelial cells that line the circulatory system. Metabolic diseases. Dysregulated metabolism disorders include type 2 diabetes, obesity, and insulin resistance. Sirtuins are involved in metabolism through their effects on mitochondria and ATP, so it's not surprising that they benefit metabolic diseases. Many of the sirtuins, specifically SIRT1, have been studied for their ability to suppress inflammation, which is a root cause of many chronic diseases. Humans with reduced SIRT1 expression tend to have increased glucose intolerance, insulin resistance, and metabolic syndrome. In diabetic mice, administering NMN enhanced insulin sensitivity and glucose tolerance. In a study published in Cell Metabolism, the authors indicate that SIRT1 was likely the primary mechanism through which these beneficial effects were seen. Neurodegenerative diseases. Neurodegenerative diseases like Alzheimer's disease, dementia, and Parkinson's disease have also been mitigated by sirtuin activation or upregulation. SIRT1 is expressed in the hippocampus neurons, the primary center associated with memory and learning in the brain. In a July 2010 study published in the Journal of Neuroscience, mice who did not have SIRT1 had impaired cognition, memory, and synaptic plasticity. In humans, a decrease in SIRT1 levels in the brain of patients with Alzheimer's was paralleled with the stage and duration of the disease. The study also found that the greater SIRT1 levels were linked to a reduction in the time of Alzheimer's related symptoms and an increase in brain weight. Here are some factors that may increase NAD+ levels: fasting, caloric restriction, exercise, saunas, nicotinamide riboside, ketosis beta hydroxybutyrate, fructose, AMPK activation. AMPK increases SIRT1 activity by increasing cellular NAD+ levels. In conclusion, in recent years, solid experimental evidence has been accumulating, supporting the efficacy of such supervitamins in preventing age-related diseases. These molecules can now be safely used to formulate new dietary supplements, possibly prolonging lifespan. Combining a customized supplementation of natural molecules with personalized physical training and nutritional regimen will be the future strategy to improve sirtuin production and maximize its benefits. Sirtuins are proteins expressed by SIRT genes that perform an enzymatic activity stimulating chemical reactions essential for the body. Increasing knowledge and research regarding the benefits of sirtuins is ongoing, although their function has been ascertained by several studies. I hope this has been an informative video to help you become the best and healthiest version of you. Don't forget to subscribe to UCanBeHealthier TV channel. I look forward to seeing you next time. Bye for now.